So a very good morning to all of you, and immense thanks to Dr. Shrikhande for inviting me for this workshop. It reminds me of the old days when we were associated with her right since 2010. It's only after COVID that she has made her center very well professionally independent, and now she's carrying on with renewed vigor. Now I would like to talk to you about basics on semen analysis. Now you all will be puzzled that today when we are doing CRISPR, when we are doing the biopsies, when we are doing PGD, PGT, why should we bother about the simple sperm as such? Because still today, nobody has actually estimated the strength and the power behind the most motile and the most smallest cell of the body, that's the sperm. Because ultimately what is embryology? Embryology is nothing but a fusion between the two cells of the body. The largest and the most sensitive cell, that's the oocyte, and the smallest and the most motile cell is the sperm. And what we are doing subsequently is monitoring of the development of the embryo in the same way as we monitor the milestone of a child. We grade the developmental pattern in the hours. 18 to 20 hours fertilization, 25 to 27 2 cell, 38 to 42 4 cell, 48 to 52 6 cell plus, then morulation, cavitation, expanding, etc., etc. So if we start the procedure on Monday, by Friday evening or by Saturday morning, we would be seeing is a well-developed expanded or a hatching blastocyst. Now this procedure would have been properly done if and only if the genome of both the gametes is ideal. Because today all the techniques, methods, investigations, what we are doing is just based on sighting, shape, size, structure and morphology. There is no parameter which actually tackles the mole biological content of the gamete, except one test, that's the DNA fragmentation, which has taken up by storm, but unfortunately it also has its own hiccups. I know it is advocated left, right and center by everybody, but it is also not an indicator. So taking into consideration the mole biological aspect, it is very difficult for us. And an advantage of the sperm part is what? You won't believe till today, WHO was not recognizing IUI as a technique for ART methods. The reason why? Usually ART means handling of both the gametes from vivo to vitro. Whereas in IUI what we are doing is, we are handling, tackling, washing, playing with the sperm only. Oocyte, monitoring, sonography, substantiated by the hormonal changes. So basically in this talk, I would just like to cherry pick certain points of what exactly you should be aware of. Don't be apprehensive of, because I've always seen one thing, that whenever labs are set up, IVF, ICSI, now the trend is focusing all towards ICSI only. Now the sad part is, when the union takes place in a natural condition like an IUI or an IVF, they fuse only when both the gametes per se are excellent in all the aspects. That's the reason in severe sperm problems like OAT or mild teratoestheno, where the embryologist has to select the sperm, and believe me, our human eyes are not capable of selecting the best sperm. So this next step, when we inject it, we are bypassing the zona reaction, we are mechanically injecting the sperm. So obviously we are bound to get a fertilization, but the problem and the real hiccups start later on. How ideal is the subsequent cleavage pattern, and how excellent or optimal will be the implantation. Anyway, let's put that aside. So why we are trying to cherry pick on the sperm as such? Because a good analysis or a good investigation will really help the patient adopt to simpler techniques, cheaper techniques, because you all must be aware the biggest hurdle in ART treatment is the cost factor, vis-a-vis the less success rate. Now success rate also is a 3-step success rate: your beta HCG, your cardiac activity, and the last is your take home baby. And believe me, there is a mile of a difference between all of those three. So there is a little bit of confusion. So anyway, we'll just start this. Now a peep into history. Anyway, now sperm were seen as first as 17th century, but till that time, there was no standardized protocol or a format. Semen used to be observed. Now, when we observe a semen sample, some people were saying, oh, I'm seeing something moving, spermatozoa. Some say, no, there are of more in this sample, less in this sample. So the count came into prominence. The shape, subsequently morphology came into prominence. Concentration, some more, some less. So there are various parameters were all amalgamated together. And in the end, all they were pulled together in a sequential pattern, and thus slowly arose the formation of semen analysis standardized reports. Now basically, semen analysis or spermiogram, it will tell us everything about the sperm. The external features, the shape, size, dynamics, kinetomatics, etc., etc. And all have been formulated in our WHO reports, which are continuously been generated every 3 or 4 years. They will tell us everything A to Z of the sperm, right from the pluses and minuses. Of course, the variations keep on going on as per the investigations progress. Now, important thing, a semen analysis will tell you everything except the fertilizing potential of the sperm. So that's the challenge, that at times when we see huge count, fantastic motility, but still fertilization is not proper. A gentleman suffering from diabetes, count and motility will be fantastic, but still fertilization failure is there. So there are many a milestone which are to be yet to be investigated as such. So remember, all that glitters isn't gold as such. So the very fact that you cite a sperm doesn't mean it is capable of fertilization. If an egg fertilizes, doesn't mean it is capable of cleavage. If an egg develops still a blastocyst, doesn't mean it's capable of implantation. And if it implants, doesn't mean it is capable of carrying it till the term. So these are all the lacunae, because still today in embryology, there is a big enigma. Implantation and fertilization. The mechanics and the dynamics of still are yet to be deeply understood. Now, prior to the formation or prior to the investigation of our radioimmunoassays, initially what we used to do? We used to do the biopsies. Fix in the Bowen's fluid, check them up, do the biopsy score, and then make a judgment as to what extent the problem lies. Complete spermatogenesis, arrested at spermatids, spermatogonia, peritubular shrinkage, blah, 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 8 to 10 parameters were there. But now with the hormones available at the click of a mouse and the reports available within 7 to 8 hours, we can immediately make a judgment as to what is supposedly wrong with the patient. And accordingly, we can opt for the treatment. Now, these are all the WHO reference guidelines down the years. Now, you can see majority of the parameters are the same, except the morphology, the normal forms. 50, 30, 14, and now it has dipped to just 4. Now this shows that as far as sperm quality, productivity and functionality is concerned, morphology plays a very important part. Of course, kinetics do play a role. Normal, IUI, IVF, kinetics are very important. And believe me, when we do even ICSI, even in a severe OAT sample, the basic parameter what we should see is the sperm twitching. Howsoever less number of sperm it might be, a twitching sperm means its membrane integrity is intact and the viability is intact. Because unless and until any cell is not viable, it will not be functional. That's the basics of cell biology. Because sperm is also an animal cell. It has a plasma membrane, trilamellar protein lipid, fluid, sensitive to change, temperature, pressure, osmotic stress. Correct? So the intactness of the membrane, motility, subsequently comes is the morphology. So all these three parameters amalgamated together will tell you the optimal function of the sperm. Now these are the booklets. You can download them from the nets. This is basically a general morphometry of the sperm, the Strix criteria, which was by Thinnus Kruger and Daniel Franken from Tyberger Institute of South Africa, from whom the Kruger's criteria is named as such. And the another one is the WHO criteria. Literally both are the same, not much of a difference. Now, morphometry and kinetics. Well, we all know a sperm has to be motile, functionally motile. The reason is functionally motile, yes, but what is it gonna deliver post-motility? The sperm acrosome, like in many of the mammals, if you see mole biologically, the DNA fraction is condensed to more than 6 to 7 times in a tightly packed. That's why when you see a sperm acrosome, 85 to 90% is occupied by the nucleus. There is no cytoplasm in the sperm acrosome, if you have seen it carefully. Now, when it decondenses in the ooplasm of the oocyte, it bursts open, it liberates its DNA content. So there lies the precursor step of the formation of the zygote into a metaphase 2 oocyte. So this tells us how the quality of the sperm does play a matter, or does play a very important role. Because in a sperm there are 3 parts: head, midpiece and tail. All have separate functions. But for a complete functioning of the sperm, all the 3 should function in unanimity. Mitochondria powerhouse, what does it do? Gets activated by the heat, releases ATP, transferred to the tail. Tail is a locomotory organ, whiplash motion, dynamics, effective stroke, recovery stroke, sperm propels forward. The aerodynamic shape of the head, the pyriform shape, it enhances motility. In a normal condition, estrogenic mucus with fantastic spin bar kit will help the sperm to propel further all the way till the tubes. Hostile mucus will impair the progress. In a lab, your media, equilibration, incubation, and the pH. That's why they are so sensitive. So all the 3 parts amalgamated together play a very important role as far as the motility dynamics are concerned and as far as the mole biological dynamics are concerned. Now in this, left side is just the normal nomenclature. The mid is just the 3 parts. And the extreme left are the mole biological aspects, which we won't touch it as of now. Now, what does a report include? A good semen report includes all these parameters. Right from the time how many days abstinence is kept, the completeness or the intactness of the sample which is collected, all the count, motility, concentration, yawing, peripheral inclusions, nature of the cytoplasm, pH, etc., etc. You name it and you have it. Now you would say, why the hell are these important? If you were to send a patient to an andrologist surgeon for further investigations, these parameters prove of vital importance. They may look innocuous to us: odor, color, texture, etc., etc. Now, these are some diseases which can affect spermatogenesis. There may be genetic disorders, hormonal imbalances, other conditions, autoimmune disorders, infections. So a proper history taking is a must. And many a times, the concept in our culture, Indian culture, is that infertility is concerned, the male will always say, nothing is wrong with me, everything is a problem in my wife only. So that's the reason many a times they are hesitant in even doing a semen analysis. A wife undergoes brutal treatments for 2-3 cycles only to find that the husband was as good as azoospermic. The trouble is, he has never taken the trouble to investigate himself. And the most sad part is, those infertility specialists have done the biggest flaw, because whenever an infertile couple comes, the first thing is do a routine semen analysis, irrespective of the abstinence or whatever it is. Once you see a good quality of the semen produced, it's a green signal from the male part. Of course, still the fertilization potential is in a limbo. We are not sure about that. But at least one part seems to be covered. Now, These are the medications which can impair spermatogenesis. And another are occupation hazards. Now certain occupation hazards are what I would say iatrogenic. A normal male, but due to certain nuances, like wearing tight clothes, the tight jeans for hours together in a day, sitting cross-legged with hot laptops on the laps after going home exhausted, taking hot sauna baths at 80 to 70 degrees water, lying there for hours together. This is heat-induced spermatogenic problems. Smoking, extra tobacco contamination, saturation. So nicotine concentration goes high, induces lazy cilia syndrome. So these are all induced trauma as such, other than the naturally occurring like spermatogenic failure, testicular failure, etc., etc. So this is how we categorize. Azoospermia, aspermia, difference. Azoospermia, no sperm in the semen. Aspermia, no semen itself. Now we come to the first part, that is sample collection. Now many of us just think that sample collection means whatever the gentleman collects, they see something present, absent, they just write a report and send it off. No. Sample collection is the key to understand the evaluation of male infertility. It is not like blood collection where we just push in a hypodermic, pull out the blood, send it into an auto-analyzer and get the printed report. There are multiple factors which will affect the collection, because a gentleman on the day of a collection, when he is told on the day of the procedure, he will undergo various sort of tensions, inhibitions, situational anxiety, collection anxiety, environmental anxiety, result-oriented anxiety, financial anxiety. So all this work negative. We have seen on just the first day when a couple comes, you tell him to collect a sample, it's like a white coat hypertension. He collects it beautifully irrespective of the abstinence. But on the day of the procedure, many a times they go temporary azoospermic or aspermic. This is because of the stress. So what we have to do is, we have to try to minimize the stress. We have to induce confidence into him, because such arbitrarily reported samples will completely alter the way of treatment. He may be normospermic, but on that day, because of certain anxiety, he may show a very pathetic report, and we have to report what we see. So in such cases, when there will be fluctuative counts, what we can do is, we can cryo-freeze the sample as a backup in case of an emergency. So there are lots of remedies available. Collection methods: masturbation, coitus interruptus, penile vibratory syndrome, and electroejaculation. Masturbation is the best. Coitus interruptus, we have found that at least in 90% of the time, the first squirt of the ejaculate is missed. And more than 40 to 45% of the best quality of the sperm are revealed in the first squirt of the ejaculate. Patient thinks otherwise. हाँ सर, शुरुआत का हिस्सा गिर गया, बाद का मैंने बरोबर सही तरह से कलेक्ट किया है. Now the problem is we have to counsel the patient accordingly, because we haven't counseled him properly. Right? So masturbation according to me, but then there are certain inhibitions. Some can't, believe me, 5 to 7% of the people haven't masturbated in their lives. They don't know what masturbation is. Some can collect by masturbation, some can't ejaculate during an intercourse, and vice versa. So we have to ask them. Some have got a location anxiety. Sir, यहाँ पर नहीं होगा, हम घर से ही collect करके लेके आएंगे. Agreed. No, घर से collect करके लेके आओ, but then there is a problem of sample switching. We had found that in many cases. They used to substitute somebody else's sample. So then we had to form a format that we, Mr. and Mrs. blah blah blah, have collected the sample at home to be utilized in this lab for the procedure on blah blah date, signed by both the husband and the wife together. Another thing, home collection. Many times we always see the problem. Samples are collected, but for the procedure they are not processed immediately. They are kept standing on the laminar flow for hours together. The more the time the sperm gets exposed to the seminal plasma, the higher and faster will be the dip in its fertilizing capacity. So if a sample is collected, it is not processed within half an hour, 85 to 90% of the fertilizing capacity of the sperm is lost because of the seminal plasma induced toxicity. So seminal plasma post-ejaculate is not a blessing. It is a blessing in a normal condition where it insulates the sperm against the acidic nature of the female secretions. Because the seminal plasma basically is alkaline, basic. So in a collection, these are all the stress factors a male has to face. Now touch wood, this slides and all the investigations and formulation was done thanks between a good collaboration between me and Dr. Rupin Shah, who was a very good andrologist, who was with me for years, or rather I was with him for years together. Now this is a good collection room, it should be. A homely ambience. Don't substitute it for a collection room because sorry to say 7 out of 10 collection rooms which I have seen are pathetic toilets. They are nauseating, they are a turn-off factor. So forget an ejaculation, a male won't even have an erection. And second thing, the biggest apprehension a male faces during collection is what? Somebody knocking at the door. Assure him that for the first half an hour, nobody will knock at the door. But if you prolong by at least 45 minutes, I myself will disturb you. Because if a gentleman can't collect by masturbation in the first 40 minutes, he will never be able to collect a sample. Howsoever normal he might be otherwise. And then you can't do a tisa-pisa also. The reason why? The musculature, the epididymis will go flaccid. So there will be no way to put in the needle and do the aspiration. You got my point. So these are the nuances. If the couple or if the gentleman is counseled from first, if you know what the apprehensions are, maybe we can tell him. Because in ART, every treatment needs reassurance, faith, and confidence. Then and then only the patient will trust you. So that's the reason when even a failed cycle patient will walk back to you if that couple is happy with your treatment pattern and the way you counsel and you treat them subsequently. Now, the abstinence period prior to collection, the gap. The reason is it helps replenish the sperm battalion in your accessories, especially the epididymis, the chorda, which is the storage place. WHO is recommending 5 to 7, but lately it is found that 2 to 3 days is a very nice period. Now when we explain this to the patient, that bhaiya abstinence rakhne se tumhare sperm ki sankhya बढ़ sakti hai. So what do? They quietly keep a lengthy abstinence of 10 days. So we can immediately make out. The concentration will be quite high. But the sperm will become senescent. The motility will be compromised. They look as if they are pooled together in a vacuum, in a condensed pack. So again the kinetics will be affected. And we don't know exactly this pooling in chorda will increase the ROS, the reactive oxygen species, reactive nitrogen species, and will promulgate dip in the fertilizing potential. So tell them the pros and cons of a larger abstinence period and a zero abstinence period. So tell them that when you are reporting a semen sample to you in case of an emergence or an uncontrollable nightfall you have, please document it. Because if I see a sample, oh just 5 million, but he had a nightfall the same night prior. So it cannot be justified as such because you remember spermatogenic cycle is 68 to 72 days long. So by the time the new battalion or the new progeny of the sperm comes, settles itself in the chorda, ejaculates, it should have some substantial quantity to be shown in the ejaculate subsequently. So what are the issues with a lengthy abstinence? When a person is abstinence for a very long time, these are the things which may happen. Okay? Then some have got collection issue. The best is you can use as a handheld vibrator. It's just a body massager. What it does is, when placed on the lower part of an erect penis, the shaft, which is richly innervated, you start the machine, it just vibrates, gives an orgasmic effect, and a patient comes into collection. They are very happy with this procedure. So some people even buy that machine. And please tell them to practice this in a peri-ovulatory period. Because it's then and then they will be acclimatized to do so on the day of the procedure as such. Because they have never experienced this before. So they really get a nice feeling, and the very fact that when you tell them that by this procedure you will get a lovely collection of the sperm to be used for your procedure, you can even do an IUI. Or you can even do in very class 2, class 3 cities or villages where we go. It is just stimulation and timed intercourse which has given us the result. So it is not like that any fertile people who have not been able to conceive, only IVF ICSI is the best alternative. No. These are also the cheaper modes. Failing to do so, you can subsequently step up the treatment pattern. Like say, 4 cycles of IUI failure, then definitely it's an IVF, depending upon the indications. The next would be electroejaculation. Availability or to produce the sperm, because as long as a male produces sperm, you cannot opt for a donor. That's not the case as of now. Now in case of severe OAT, certain sperm sample will not be shown in all the samples, but suddenly a third or the fourth ejaculated sample will show presence of few sperm. So this is called as sample skipping. So by the procedure of cryptozoospermia detection, we can find out the presence of sperm in the pellet. You collect the sample, spin the sample, check the pellet. So that is the case of cryptozoospermia or hidden sperm. Now many a times, very chronic OAT, or a person is paraplegic, incapable of movement waist down, restricted to a wheelchair. This electroejaculation really helps retrieve a good number of sperm depending upon the spermatogenic capacity of the testes. This is an anal probe done under observation, under GA. So what is done is, it is inserted, slowly the current is switched on, mimics again the orgasmic effect, you collect the sample. You can't repeat the process off and on. So whatever you collect, you have to store it. They are very sensitive, but somehow, even though the count is huge, unfortunately the motility in electroejaculated sample is always poor. So the ICSI is the best alternative. Okay? These are certain sample collection tips and tricks, which everybody has to follow. These have been noted just from the guidelines. I have just kept them in a bulleted form. Most important, abstinence period and the container. Believe me, ideally glass was used because glass is the most bio-friendly material for any biological fluid. We must be aware we had saline bottles of glass, we had hypodermics of glass, even we had Petri plates for culture in glass, but glass cannot be thrown away after one use. In the process of cleaning, washing, sterilizing, autoclaving, drying was huge and cumbersome. So then developed the plastics. But these plastics are not cheap plastics or simple plastics, agree. All disposables in ART are made from polystyrene, which is a polymer of a plastic. They also release VOCs and COCs, or volatile organic components and chemical organic components. But when these are manufactured, care is taken to see that the reprotoxicity for the gametes is absolutely zero. The way they are packed, auto-sterilized either by gamma radiation or by ETO, whether it is a sperm, whether it is a follicle aspirate, or whether it's a media, the QA/QC of the material has to be excellent. So please, good quality, large mouth containers, because we want to collect the whole sample. Years ago, we used to follow a method, what we call it as split ejaculate. Part 1 and part 2, but it was very difficult to judge between the two. Okay, spillage, as I told you, and added assistance with the last person. Now, post-observation, when you collect a sample in an infertility clinic, maybe a junior embryologist, any person will be having, just tell her to observe a drop of the semen under your microscope, which will be having 10, 40 and 100x objectives. So you will note the volume, you will note the nature, whether it is normal or viscous. We will note the pH, because volume and pH coexist. Then the color, the appearance, the odor, the number of sperm saturated inside the droplet, the way they are moving, and peripheral cellular inclusions. Whether it's a pus cell, premature cell, phagocytotic cell, cast, etc., etc. Omit this, yeah. So this is what exactly it is. Secretions versus the pH. 85% of the secretion is thanks to the seminal vesicle, which is always alkaline. Rest, prostate, acidic. Least bulbourethral, which is also acidic. So if a gentleman collects just 0.1 to 0.2 ml of glassy, transparent ejaculate with no sperm and the pH is acidic, he has just collected the initial bulbourethral secretion, which is just like a lubricating phase. Then comes a little bit of more when the prostatic secretion pumps in. This which happens because of the nervous control, because all the accessories are richly nervous control, because when we get excited or aroused in an orgasmic phase, the tactile, auditory, sensory stimulations all work together. So the pumping action from the chorda pushes the sperm via the vas deferens through the accessory gland complex, first mixed by the prostatic secretion, and in last, the seminal vesicles contract and give it the maximum volume. So the maximum volume comes at the last part of the ejaculate, and that is always alkaline. So pH and the volume is very important for an andrologist to know what the problem is. Okay? So simple looking parameters. Now there on the left side, you see all the contents of a semen. Semen is a plethora of flora and fauna. So that's the reason raw semen or neat semen is never taken into a culture laboratory. Andrology lab is separate, incubators are separate, but washed or capacitated sperm can be taken into the lab as such. So prior to recruiting any person for any procedure, we used to do a few tests. Normal semen analysis, a trial swim up, a 24-hour survival test, to see whether the seminal plasma was clean, it was not hostile or not infected. And subsequently, what was the sustaining capacity of the sperm post 24 hours? That was very important. So, many a times we get lots of pus cells. Now the patient might be suffering from lower UTI and the patient might be suffering from prostatitis. The symptoms of both are the same. Do a routine urine analysis and do a routine semen analysis. Pus cells in the semen exceedingly 4 to 5 per high power field. It may be prostatitis. In the urine, it may be UTI. Sometimes we get large volumes, 7, 8, 9 ml. Very watery, yellowish, turgid. Either a prolonged abstinence or seminal vesiculitis, infection of the seminal vesicles. Now, seminal vesicle and prostate, both are secretory glands. Lumen, lined by tall columnar epithelial striated secretory glands. So whenever there is an infection, they produce this secretion in excess to wash off the infection, as a result of which the volume becomes large. But the 24-hour survival of these samples will give you severe contamination the next day. So by chance, if they are inseminated in an IVF dish or they are done in an IUI, the patient will be having crossover infection into the uterus as such. Or the IVF wells will develop contamination the very next day. So please recruit them properly, check the 24-hour survival, and then you can prolong your treatment as such, or keep them under heavy antibiotics. Worst, if it is not improving, go for a culture. But 35 to 40% of the time, even cultures come positive. And what the remedy we say? Heavy dose of antibiotics, do a test, 24-hour survival, and then you can carry on. So this in a nutshell, it tells you the fluid components in a ejaculate. Seminal vesicular, what do they do? The bladder, vas deferens, etc., etc. So, a human semen is a conglomerate of two fractions. One consisting of the cellular fraction and one consisting of the acellular fraction, which is just the liquid part which comprises of the seminal plasma. Now it is this seminal plasma which we eradicate during the process of an IUI. We clear the sperm of the attached seminal plasma, so we say we are washing it. And when we wash it, what do? We capacitate. We enhance the capacity of its fertilization. We activate it we increase the acrosomal content of calcium ion prolongation. We remove those membranes from it, especially the glycoprotein membrane, which improves its motility. Correct? Proper temperature and media enhances the functionality of the kinetics. So all put together, we are forming it in such a way that it goes actively towards the well-receptive cumulus complex of a metaphase 2 oocyte, which is going to meet either in vivo in an IUI or in vitro in an IVF procedure. You got my point? So what we are doing is we are mimicking the estrogenic mucus in vivo thanks to the media in vitro. So that will tell you the necessity of good media and good equipments, which subsequently we'll be taking in the lab procedures. Now these are simple volume related points which are to be noted as such. Definitely age related, concentration wise, habituate, life, what you call it as a life pattern, your nasty habits. pH and its importance. Well, internally, sperm have to protect themselves against the acidic pH of the wife's secretions. Acidity high, insulation of the sperm by its seminal plasma alkaline nature is poor, acidic denaturation of the acrosome. So the functionality of the DNA dips. In vivo, now in vitro, your media is not properly equilibrated or incubated. The intra and the extracellular pH varies. So obviously again the functionality of the DNA in the acrosome will be poor irrespective of the kinetomatics. So that's the reason whenever such things fail, in an IVF which is a natural selection, we get FERT failure. People always scrib, अरे IVF में तो FERT failure होता है, मगर क्यों होता है वो आप देखो. You are not processing the sperm properly. You are not capacitating it. You are not eliminating the seminal plasma. You are not giving it temperature and osmolarity consciousness. But if in an IVF procedure, you get 75 to 80% of fertilization, I am sure your embryos would be par excellent and you are mimicking the uterine environment in your laboratory. Because it's a natural selection. So I always term IVF is like love marriage, ICSI is like an arranged marriage. Because whatever we get of sperm, we have to inject forcibly. Bad egg, good sperm. Bad sperm, good eggs. Both bad and an unexplained cause is an indication for ICSI as such. Now this is the causes. Alkaline and acidic. Now liquefaction. Now we all know 80% of the semen is from the seminal vesicle. It comes as a coagulum. It is thick. It needs to be broken down. And it is broken down by the secretion of the prostate, which contains prostatic specific antigen. Now if the prostate per se is infected, unfortunately this breakdown is not possible, so we get the long thread-like happenings in semen, contrary to our mucus, which is a spin bar kit, means it's a lovely expansion. The longer the spin bar kit in a mucus, the more receptive or estrogenic is the mucus. The more brittle or the more hostile the mucus, it will be like a barrier. But in a sperm or in a semen, the longer the threading, the poorer the quality of the semen. So this viscosity has to be nullified. If it is a procedure for an IUI, don't wait for the semen to liquefy on its own and then you write down liquefaction complete within 1 hour. No, but by that time the chemical damage to the sperm acrosome is already done. You have to manually liquefy the sperm by needling it to a 20 gauge needle. Now don't ask me, please, 20 gauge needle, oh my gosh, sperm are so tiny, they will be damaged by the needle diameter. No. Your ICSI needle in which you inject is 100 times thinner than that 20 gauge needle. So sperm damage is not there. It's a manner in which you liquefy it. But if you are doing an analysis, wait and watch how long it takes to liquefy. So according to that, you can judge the intensity of the infection which is induced into your seminal plasma. This is how we do. Unit is centipoise for a semen. 6 to 7 centipoise is the unit of viscosity. Odor, well, usually it is seminiferous, but if there is a severe infection or an STD infection or a bacterial infection, it's a severe foul smelling. Correct? Color, red mostly is due to hemospermia. Bladder infection, prostatic carcinoma, prostatic infection, or certain STDs, or certain minor changes which can be induced by those occupational hazards. Now, most important is the count. Now, whenever we ask a report, what do we always say? So, uska count to bahut badhiya, 80 million. And then slowly comes the punchline. But motility is only 10%. So, out of an 80 million count and 10% motility, the functionally motile sperm are just 8 million. And that also can be broken up into the subsequent characteristics. Grade A, grade B, grade C. Now the grade patterns have been changing a lot. First it was like a number, 0, 1, 2, 3, 4. But then it was discarded because some were giving in ascending order, some were giving in descending order. Then came the text pattern. Active, moderate and sluggish. Even that was a little subjective. So now WHO has considered only two patterns, progressive and non-progressive. Because even your semi-progressive sperm, post good wash, can turn into active progressive ones. Second thing, always register count as million per ml, million per ejaculate, and the total motile sperm count. Like how I said, 80 million count, 10% motility, so the total motile sperm count is 8 million. That will tell you the actual number of functionally viable sperm in a semen sample. Don't get carried away by the exorbitant number of that initial 80 million as such. Correct? That's very, very important. So this is the motility part which I just discussed. Now, for that, the basic and the most important thing which you need is a very good bench microscope. See, don't make your lab a plethora of gadgets of the latest technique and the latest updated model. Be very simplistic in selecting your gadgets. Essential to the functionality, but that gadget should be excellent. Don't buy an X gadget because my neighbor has it. Oh, that's in the market, so I have to have it. These are mere add-ons. Believe me, these add-ons are doing nothing else except drilling a hole in your pocket. And ultimately the patient has to pay for it. And the high price will again prevent the patients from walking into your laboratory. So this, a very good bench microscope, either it can be a trinocular or a binocular, all the four types of magnifications, 10, 20, 40 and 100, good optics. So proper resolution of the sperm is always done in the andrology lab by a good andrologist on this microscope. Then this is what we call as the grading. Grade A, straight uniform motion, B a little haphazard, and C total wavered. So progressive motility, so therefore there are two issues. Linearity and progression. A linear sperm is always good progressive, but a fast progressive sperm can never be always linear. Just like I always say that all infertility specialists are gynacs, but all gynacs are not infertility specialists. There is a spatial way of modulation of the course, number of parameters you have to learn right from patient recruitment, folliculogenesis, process implementation, etc., etc. This is a diagrammatic pattern of the progressive versus non-progressive. Why? Because the linearity along with the angle of yawing. Now what is yawing? A sperm always goes like this. So yawing is a phenomena in which a body goes in a straight line, but one part changes the direction or a plane. Now this yawing is very important when 25 to 30,000 capacitated sperm in an activated version will approach the cumulus cells of a metaphase 2 site, activated. So there is a lock and key mechanism between the two, a natural force of attraction. So this yawing with the linearity and the motility will approach them. The moment the acrosome touches the cumulus, what's the first reaction? It's the highest reaction or the hyaluronidase. They will be liberated. The peripheral cells are digested. One lucky sperm goes, does the membrane reaction, The moment the membrane reaction is done, cortical reaction takes place around the oocyte, prevents polyspermia, and that one lucky Charlie goes in, fertilizes, decondenses, and forms the zygote. So this is the importance of motility, kinetics, and morphology in favoring optimal fertilization. Now this is what I would term. We look like a sperm is traveling in a straight line. No, but it always zigzags a little moment. That's called an angle of lateral amplitude. And below are just the speed representations of different type of sperm in the motility concept. So initially they were graded like this. This was yawing, which I just discussed. Now when we count them, don't make it totally subjective. Don't estimate. Because sometimes when the count dips below 10, you have to be a little precise. And at that time, these gadgets like the sperm counting chambers are a blessing. But pay respect to the gadget. Now, whenever we do a sperm count in a macular, the first and the foremost thing you should do is make the sperm non-motile. Because if they are not made non-motile and they are placed on the grid, they will jump from one square to another, making your 5 million count look like 15 million. For motility, it is excellent. The reason why, it's not showing over. The reason why is with this distance between the base and the cover slip is just 10 microns. So it will accommodate only a monolayer of sperm. Simple testing, पूरा 10 minute भी लगता नहीं spot testing के लिए. But the spot testing, if done properly, will really give you a total idea of what the sperm parameter is. And remember one thing. One sample is not a yardstick to judge the androgenicity of a male. First sample will come 2 million. You'll say, chala, XC ka patient aa gaya. No, not like that. Call him again after 15 days with a gap of 2 days prior to each collection. Collect the sample 3 times. It may be first 5 million, 45 million, 30 million. Or it may be vice versa. First sample 60 million. Second time when he comes 8 million. But you know, already he has given a 50 million count, so he is capable of normal spermatogenesis. So don't arbitrarily judge as such. Take a little average and always collect the sample in the morning hours, because if you call the gentleman during the evening time, stressed out, tired, God knows whatever mental stress they had at the place of work, the anxiousness to reach the lab within a speculated time, all these work against the collection methodologies and the outcome, because a person as far as an ejaculate is concerned has got no control, right? Or neither the lab people have the control over it. Like as I told you, for blood, if you don't get a vein over here, you take a butterfly needle and take it from here. We have got the options open, but in that case what? And in the first collection, when he gets a poor count, don't screw up his morals by saying, count is low, boss. He will carry that apprehension for the rest of the treatment time. He will always think, my sperm is bad, I am never ever going to father a child. So it is a mental inhibition which he will get. Tell him it fluctuates. Explain to him the reason. You got my point. Now this is how we see them in 20x and 10x. So a macular or a counting chamber is really a blessing because it helps you instant evaluation within a minute. So alternative to counting was what? Our hemocytometer. Over there what we used to use? Semen diluting fluid. And what does semen diluting fluid contain? Dilute formalin to make the sperm non-motile. The idea is, suppose if we were all wearing the same clothes, same colored clothes, and we were just running around in the room, a person from outside would have been able to count how many are we. But if you are standing still at localized place, we can easily count them. So that's the logic behind it. Now comes the important part. The techniques of washing. Elimination of seminal plasma. Capacitating them. Activating them. Making them ready. Mimicking the conditions similar to a good estrogenic mucus. Sperm recovery. So, in case of samples, again we have to classify them. Normospermic, moderate, or severe OAT, oligo, astheno, terato. Well, all the parameters are on the dipping side. So what this method tells you, it is just like a natural selection. well migration of the sample which have been condensed by centrifugation, subsequently liberating up in the media, and we collect them. So this is the principle, it's a gold standard for normospermic. But here, there are two steps to be done very carefully. Spinning them twice. So it's the first thing is, we dilute the sample. 1:2. The more diluting the seminal plasma, the better would be the pellet. It's like an example, I give you two test tubes, glycerin and sand and water and sand, you spin them, which will form a pellet faster? Water and sand. Why? Density is less, buoyancy is more in case of glycerin. The same is the case. You dilute the seminal plasma, let the supernatant be discarded. In a second spin, again wash it with 1 ml of this media, which should be always at 37, because sperm preparation media is a salt-based substrate with a little bit of a buffer that is HIPPIs. So always do plain incubation. Do not ever do CO2 equilibration if it's your embryology lab. So all gametes which are individually handled, like OCCs during aspiration, denudation of the oocyte, sperm washing, always under HIPPIs base, because HIPPIs is a fantastic buffering agent. Now, in the last process, when we take the pellet, we layer the pellet with 1 ml of pure sperm media. Now this time it needs excellent incubation in an incubator. People always argue, अरे incubator में क्या ज़रूरत है, मैं तो hot block में रखता हूँ. Fine. In hot block, the heating is by conduction. Whereas in an incubator, the heating is by convection and very gentle. So the moment this pellet gets very gently heated, the pellet becomes soft by the layer of the media. Correct? So slowly the sperm are swimming up. They are liberated because now the centripetal force acts onto it. Initially it was the centrifugal force which compacted. Now there is one flaw. People spin the sperm sample at various RPMs as per their whims and fancies. This is prone to membrane damage. Now all foreign books will have G force, if you have seen them. All Indian versions RPM, rounds per minute. Both are miles apart. For a sperm, 300 G is ideal, or what we say 1300 to 1500 RPM for 10 to 15 minutes. Because what happens, if the G force exceeds, there is membrane peroxidation or the membrane lysis of the sperm. This will increase the ROS. Plus the other delicate cells, the premature cell, the pus cells, they break down, they give out their contents and they coat the entire length of the sperm, increasing the ROS, dipping the fertilization capacity. So please, timing and the RPM has to be monitored. Second method, choosing the devil from the devils, OAT, oligoasthenoteratoma. They are not capable of swimming fast. They are less in number. So we have to selectively filter them. Now this is done by two steps of pushing them down through the gradient. Madam, that's mine. Pushing them down through the gradient. So we keep the semen sample. I'll show you in this next one. We keep the semen sample, two gradients. Heavier gradient and lighter gradient. Sorry, yeah, that's fine, thank you. So these should be layered one above the other. Don't mechanically mix them, because why? This interface between them is very prominent. On the top layer, not more than 1 ml of good liquefied semen. Don't overlay more, because this will screw up the fertilizing capacity of the gradients. And one more thing, these gradients have to be at room temperature. Don't take them out from the refrigerator and immediately wash it up. Fine. Spin them. You collect the aspirate down at the bottom. Take the small pellet, mix them with the media, incubate it, and do it. This is best for ICSI samples because they take care of the morphology as well as the density gradient. That's why it is called density. So as I just told you, sorry, 1 second. Yeah, this takes care of this parameter. The sperm whose acrosomal density is between 1.6 to 1.09, they are the best capacity as such. That's why it is called double density discontinuous gradient separation. So these are the general tips and tricks for sperm processing. Okay? And last but not the least, the morphology. It is the staining pattern just as you do your cervical smears. Pattern is what? Again the pH, acidic basic. Acidic nucleus is stained by basic hematoxylin. The cytoplasm, which is basic, is stained by an acidic stain. There is a color contrast. So if I was to show you this one and ask you which of sperm is looking normal, I would say only 4 is near normal because all have got some sort of an abnormality. Number 1 is a vacuolation, abaxial placement. Number 2, shape is funny. Number 3, midpiece defect. Acrosome percentage is not proper. Break over here. So if you cherry pick a sperm in OAT sample, believe me, you would find all of them improper. That's the reason when you do ICSI for OAT sample, all of them will show fertilization and early cleavage, but later on they will be arresting. Now look at this. Multiple problems. And the sad part is, these abnormal ones swim the best. So when we look at one drop, that's why we get some labs from XYZ labs, 2% abnormal because they say everything's motile and they think everything motile is normal. That's in irony. This is an pinhead, no acrosomal complex at all. They say it is because of the stress. Hypothalamo-pituitary gonadal axis. Now infertile person is always stressed out. In the hypothalamus, 2 cells, supraoptic, paraventricular, which release the gonadotropic releasing factor. Now if the patient is taking depression medicine, that whole axis is messed up and the last target organ is the Sertoli cell, in which the sperm are formed subsequently. So as long as this is under stress, the performance of the Sertoli cell is definitely going to be compromised. Different patterns. Vitality, large number of sperm, majority are non-motile. So non-motile sperm are alive, they are never dead. So we do a membrane integrity test. The ones which are pinkish in color, they are dead ones because the stain has penetrated into them. Change the lifestyle. If he's a tobacco smoker, tell him to abstain from tobacco for at least 3 to 4 months. Nasty habits, hot water baths, severe, eliminate it. There will 30 to 35% recovery. Then I think I will skip this DNA damage or what we call it as the DFI problems, single strand or double strand. These are the 3 reasons. Oxidative stress. This is reversible. Unfortunately, these 2 are not. I'll just explain one slide to you. I take the sperm, rip it apart, get the helix, get the DNA, get the protein core, rip them apart further. So these are the toroids which are ultimately affected in your DNA fragmentation. That's the reason the 3 tests, tunnel, Comet, SCSC and SCD. Now this is exactly what we are doing in our labs. We are studying as per the expansion of the acrosomal part. This makes the test very sensitive and meticulous. Equipments are huge and they cannot be done over the counter in a jiffy. If you can't interpret the test properly, please don't do it. This is just an add-on. It will have you one more ammunition in your basket to say, yeah, this might be the reason you are not conceiving. But this is not the sole reason. This is the parameters of a DFI, usually. Then these are the 4 tests in comparison. We are only doing the last one because these 3 are phenomenally costly, not possible with an individual capacity. The reasons why: age, testosterone deprivation. Of course, a man can produce semen till 75, 80, but the functionality is quite poor. Just some statistics. And my most copied slide. Yes, favorite. Anyway, that was my last slide.